So thank you all for joining this month's Armchair Botanist Forum. I'm really excited for this presentation. Our very own seed bank conservation botanist is going to be presenting today, Kay Hankins, and she's the latest employee to the research department, and it's been really awesome to see kind of the seed bank and the conservation program like really blossom and grow this year with us. So I'm going to hand it over to Kay and
Hello,
let her get
everyone.
started.
Can you hear
Thanks.
me?
Yes.
Thank you. I'm so happy to be here. Thank you for having me. Today, I'm going to be talking about conserving Texas native plant biodiversity, seed banking, biology, and research here at the Botanical Research Institute of Texas. I want to start by just providing a little bit of background about who I am. I have a bachelor's of science in biology that I earned from Sam Houston State University in 2018. I've gone on a long, strange trip between then and now. I've worked as a farm technician and ecotourism guide with organic sustainable agriculture in Captain Cook, Hawaii. I've worked as a botanical field technician in Boise, Idaho with the Chicago Botanic Garden and the Seeds of Success program, as a GIS analyst in the Mojave Desert. I earned my Master's of Science in Biology at Sam Houston State University in 2023, where I studied species distribution modeling or ecological niche modeling of invasive agricultural crop pests. I've worked as a lead botanist in Susanville, California, again with the Chicago Botanic Garden and the Seeds of Success program, and finally ending up here. And because I forgot that I included all of these really cool pictures of these places, I'm going to quickly shuffle through all of those real quick. This is the view from the deck of the farmhouse in Hawaii that I worked at. This is a lovely view of the Sawtooth Mountains in Idaho. The Mojave Desert, we've got this lovely teddy bear cholla cactus here. Beautiful sunsets, always a lovely time. And Northern California, a very beautiful place as well. So just a little bit of background about the Botanical Research Institute of Texas, and many people don't know we actually have this conservation seed bank here. So I want to give a little bit of background about when that was established. So the institution itself was first established in 1987 with a very noble mission of exploring, discovering, and engaging the public. We explore critically important world of plants, collaborate to discover the role they play in our cultural and natural environments, and engage people to conserve nature and improve the human experience. So you can imagine why I was super excited to come on board here at the Botanical Research Institute of Texas in the Fort Worth Botanic Garden. We have a number of different arms, including the Phycology Herbarium, Research and Conservation, the BRIT Library, and the BRIT Press. And as of 2019, we also established the Conservation Seed Bank. And just a little bit of background, if you didn't already know, we merged with the Fort Worth Botanic Garden as part of a management agreement in 2020. So those two things happened very close together. So what does a seed bank conservation botanist actually even do? Well, we survey and monitor sensitive native plant populations, rescue and collect seeds from stable and at-risk population sites, conserve native plants through the long-term storage, curation, and research of native plant seeds, and research the biology, germination, and longevity of the seeds that we collect. And here at the BRIT Conservation Seed Bank, our primary mission is to collect, preserve, and research sensitive Texas native plant biodiversity through the use of the seeds that we collect. And I always like to include a number of different pictures because often when you mention the word seed bank, people often think of this image that you see on the upper left, Svalbard Global Seed Vault in Norway. And you may have heard of it referred to as the kind of global doomsday vault. And that's because they specialize in housing a number of wild crop relatives and cultivar species of agricultural crops that we grow all over the world as a means of securing global food security. If anything were to happen around the world, whether it be geopolitical instability, natural disasters, climate change, there's some repository of the food that we grow and eat all over the world in a place that's relatively isolated and climate controlled as much as we possibly can. You may know that the accessions that are housed in this particular seed vault in Svalbard are housed under the permafrost with the objective of keeping the seeds at a relatively cool and constant temperature that they need to maintain their longevity, which I'll talk a little bit about. On the upper right, you'll see the National Laboratory for Genetic Resource Preservation. It's in Fort Collins, Colorado, here in the United States. And it also, generally speaking, specializes in preserving agricultural crops, the seeds of those cultivars and crops that we grow for food security here in the United States. But recently, they have started to expand what they call their black box collections that are also wild native plants and primarily the Seeds of Success program, housing the backup collections for the collections made as part of our National Resource Preservation, Plant Preservation program. And so those are both institutions that are massive. You can kind of see they're wearing actually parkas in this image because these seeds, this entire facility is kept at -20 degrees Celsius, which is about -4 degrees Fahrenheit. So they can only be in there for maybe 10 minutes at a time, and they wear these large parkas to actually access the accessions because they are stored at such cool temperatures. But what people often don't realize is that seed banking is actually a pretty low tech enterprise. You don't need huge sub-zero facilities or fancy, flashy buildings that are under permafrost in Norway to do this work. You can actually do it with a relatively normal stand-up freezer in a room that has relatively constant temperature and humidity and be able to call yourself a seed bank. And so here we're a relatively small organization. Again, we were established in 2019, but we're growing steadily by the year. Currently, we have over 50 different accessions, of Texas native plant species, most of which are threatened, rare, or endangered here in the state of Texas, for a total of more than 50,000 seeds conserved in our seed bank from over 25 of those sensitive plant species. So it's pretty cool, and you can do it with a relatively low budget and low technology, which is excellent for a lot of nonprofit and smaller organizations when they're first getting started. So with that, why are we doing this? Well, global biodiversity is under threat due to anthropomorphic activities such as the increased industrialization and globalization of the world's economy, travel, and trade. And this, as you probably are aware, results in dramatic increases in annual global mean temperature, as illustrated in this graphic right here, otherwise known as climate change, right? And we see this pretty drastic increase, this upward trend in global annual mean temperature post the Industrial Revolution, right? So this has a synergistic effect, and that drives the decline of biodiversity, which often looks like habitat loss and fragmentation, the introduction of invasive species such as the emerald ash borer that's pictured here on the left, and also this kudzu vine that you will see often from your car side window if you're driving anywhere in the southeastern United States. And at present, the rate of extinction is estimated to significantly exceed historical baseline rates for many taxonomic groups. And plants are absolutely no exception in this way. This figure that you're seeing here shows that as of, when was this, 2022, roughly 40% of known named plant species that were assessed worldwide were predicted or determined to be threatened with extinction. And just a year later, the Royal Botanic Garden at Kew's State of the World's Plants and Fungi report reported that 45% of flowering plant species were at risk of extinction. And just to kind of give you an idea of a little bit what this looks like in terms of other taxonomic groups, modern day plants are estimated to be going extinct at rates many magnitudes higher than the average historical background rate of about 0.05 to 0.35 extinctions per million species per year. And what this figure down below shows with all of these different vertebrate and taxonomic groups is kind of a optimistic view and a pessimistic view of kind of annual extinctions per million species per year, right? And what I want you to take away from this is that vertebrates have a relatively similar historical background rate of extinction as plants. But under panel A, you'll see this is our optimistic rates, right? With mammals being at roughly like 1.4 extinctions per million species per year, or upwards of 2 if we're looking at our more pessimistic predictions, right? Plants are estimated to be going extinct at 18 to 26 plus extinctions per million species per year. That is anywhere from, basically 18 plus times more than what we're seeing with any other vertebrate taxonomic group. And if you look at it in the context of the figure that you're seeing here, it would be absolutely off the charts. And that's largely in part due to the fact that plants are a vast, numerous taxonomic group that includes many species. But still, it's extremely troubling how quickly they are experiencing the adverse effects of climate change and anthropomorphic activity in spite of that. And why is this a problem? Well, plants play a fundamental and foundational role in nearly every terrestrial ecosystem on the planet. And if you'll remember from basic biology or ecology, you know, uh, the loss of one plant can have a negative cascading impact throughout an entire ecosystem. And that's because, the entire ecosystem depends on those plants. And not only that, but 77% of undescribed plant species are likely threatened with extinction. And the more recently a species has been described, the more likely it is to actually be threatened. So we're actually losing biodiversity before we even know or have time to describe it and know the role that it's played in these ecosystems. And so we never actually, to go back, never actually fully know what potential services that organism provided throughout this ecosystem pyramid that you're seeing right here, or the potential uses or benefits that it could have for modern medicine, agriculture, crop improvement, etc. So it's really troubling. And that's what you're kind of seeing here is, the more that we have year description, the more recently something was described, the more likely it is to be threatened. And that has a lot to do with the being a product of rarity. If we haven't discovered or described a plant species by now, it's likely due to the fact that it's relatively rare, relatively endemic, narrowly endemic to a specific site. And so with that rarity often comes small population sizes that are extremely sensitive to perturbations in the environment. And once these species are gone, so too are their genetics, their evolutionary history, their potential and function in the ecosystem. And so that's why this is such an important thing with conservation, so that we can document, uh, preserve, and conserve all of those things before they're gone. And because me being a botanist, it's easy for me to advocate for the conservation of plant biodiversity for the sake of doing so. However, if you're not so inclined um, to seeing the value of plants and their innate value, then I offer you this, things with faces. And that is because you cannot preserve and protect native habitat for threatened or endangered fauna without addressing native plants. So to bring it back home, there's no red cockaded woodpecker without healthy old growth longleaf pine savanna. There's no greater sage grouse in the Intermountain West without high quality sagebrush steppe. And there's no desert tortoise without their preferred food sources in shade plants. And these are actually two photos that I took when I was working as a GIS analyst and biological field technician in the Mojave, conserving plants, making operational scale native seed collections for the conservation of this little tortoise here on the left. So what can we do? Now that we're bringing this full circle, we can survey and monitor sensitive native plant populations, rescue and collect seeds from stable and at-risk population sites, conserve native plants through long-term storage and curation and research, and again, research the biology, germination, longevity of those seeds that we collect. So with that, I want to talk a little bit about the actual conservation collection and seed banking process. So step one, define your goals and assemble a plan. What is possible? Because not all species can be conventionally seed banked under orthodox conditions. So for example, oaks or orchids. Both of these species cannot be stored conventionally under orthodox conditions. But what does that even mean, right? Well, orthodox seeds are those that can be dried to an internal moisture content of roughly about 12% and stored at that really cold -20 degrees Celsius or -4 degrees Fahrenheit for long periods of time and still survive. But things like oaks and orchids are considered recalcitrant and they cannot be dried to those internal moisture contents or stored at those temperatures and survive and maintain their viability. And then there's something in between, and those are called intermediate seeds, and they can be stored under orthodox storage conditions, but exhibit pretty poor longevity, roughly 5 years, as compared to orthodox seeds, which can last and retain good viability for decades. Or if you saw the recent paper that was published, maybe 1,000 years under the right conditions. Right, and they generally perform best, though, these intermediate seeds, when dried down to a relatively higher relative humidity of 45 to 65%. So those are the kind of different things that you have to plan for when you're trying to make conservation collections. What is the phenology or blooming period of your species of interest? What diagnostic features are necessary to make a positive identification in the field? uh, all these things, right? And I have a picture of Astragalus saxmaniorum. Here, that is a Texas SGCN. And that's because Astragalus specifically, I've encountered this many times, you need both the flower and the fruit to make a positive identification and ensure that the thing that you think you're collecting is actually what you're collecting, right? And sometimes that takes considerable planning ahead of time. Do you have access to stable populations large enough to support a conservation collection in any given season or multiple seasons over time without further endangering that population? And what is the maximum percentage of mature seed that we can safely sample from a population in a given year? Well, luckily for us, this work has already been done, and it was determined that 10%, no more than 10% of individuals should be sampled in a given season, and no more than 10 times out of every 90 years should that population be sampled. And that's because many different taxa, there's a diverse tolerance, unsurprisingly, to the amount of routine sampling that they can sustain without showing adverse effects to the population. And if our goal is conservation, ensuring that we're not actively harming the population that we're aiming to protect is a really important goal to meet, right? And so it was determined that 10% is kind of the sweet spot and no more than 10 times out of every 90 years. And that is as outlined in the Center for Plant Conservation's best practices for seed collection, which if you're interested in learning more about native seed collection and the best practices to do so, they have a free downloadable PDF that you can access online. So I encourage you to look more into that if you're interested. But there's always an unless, right? So unless you are performing a plant rescue for a population that is threatened with imminent and assured permanent loss or destruction. So this is a photo of some of our own folks here at FWBG-BRIT that were performing a rescue. And we collaborate with private landowners, developers, municipalities, and other organizations to rescue all available plant material at these locations. And this is the only time that we do so, and it is appropriate to do so because those sites are assured to be destroyed by development or other projects. And so that is really kind of the last chance for us to conserve the biodiversity housed in those populations. Get to know your targets. I like to liken this as to like speed dating. You need to know what they like, what's their distribution, what are their habitat requirements, where are their locations, known occurrences. You need to know all of these things before you go out into the field. And furthermore, can you get land access? Are they closely associated with any other taxa? Can you reliably distinguish them from other morphologically similar species? How will you be able to tell them apart? What materials and permits do you need? And this list goes on and on and on, right? You can never be too prepared. And I have two photos of, we have Callirrhoe scabriescula and Callirrhoe involucrata here as just an example of morphologically or closely related species that you need to be able to reliably tell apart in the field when you're doing this kind of survey work ahead of seed collection. And then next, the fun part is scouting. And this is where the rubber meets the road, or some vague approximation of a road. And so I love this photo right here because even still, the best laid plans and well-researched field plans end up looking a little bit like this, right? And this is actually a photo from a book that you can purchase called A Field Guide to Roadside Wildflowers at Full Speed by Chris Helzer. And so you get really good at identifying plants and your targets from images that look very much like this and identifying, that unique shade of green on a hillside far away. So these are sort of the skills that you acquire when you're doing field botany. Sometimes it looks like this. And you follow the map that you made, and you end up down a random 2-track that ends at a lovely vista that is also a cliff, um, and not a through road. But in the process, I will say for this particular instance, we found a population that we otherwise would not have found. So sometimes wrong turns lead to good things, but oftentimes there's a lot of getting lost involved. Not lost, but taking the scenic route for sure that's involved with scouting and field botany. But even so, nothing beats the rush of finally happening upon a suitable population of your target species or the journey that it takes to get there. So then, once you've found your population, you've identified where it is, roughly how many individuals there are there. Um, it's waiting. You have to monitor and wait for that population to go to fruit or for those seeds to mature close to the point of natural dispersal. And this takes time, and it varies significantly depending on the taxa of interest, its life history traits, and the environmental conditions at that site. And sometimes nature just has other plans, or cows, or both, right? When dispersed seeds, nothing gives my heart palpitations faster than knowing that my seeds are close to maturity, and it's a windy day. That has happened many times. Insect herbivores, not even just mammalian herbivores, but crickets, katydids, anything, um, can pose a risk to populations during this time. Um, but if all goes right, there's no cows, no wind, no insect herbivores, we get to do collection, right? And this is the meat and potatoes of the whole operation. And the fruits of our labor, 100% pun intended, right? And you can see me here at the lower left. I'm flailing a tennis racket wildly at Cercocarpus ledifolius. And I like to mention this because there's a variety of different techniques used to collect seeds. And it varies depending on the taxa of interest. And this can range from stripping to, like I said, flailing wildly in the general direction of that tree or shrub. And I can only imagine what the average passerby thinks when they see folks out in the field doing this. And luckily, we get a lot of questions and have had a lot of really fun interactions with the public. Good opportunities for outreach and education in that way, but I'm sure it does look quite quite strange to see us out there sometimes. But the method of collection also depends greatly upon the collection's intended purpose, conservation versus large scale restoration, right? Whoops. And so, with that, what is large scale for operational restoration collections? Well, this is mostly concerning the National Seeds of Success program, which I mentioned a little bit earlier, which was first established in 2001 by the US Bureau of Land Management in partnership with the Q Millennium Seed Bank Project. And this whole Seeds of Success process is just the first step in the National Native Plant Materials Development Program, which you can see kind of this wheel here on the left, the lower left, that is roughly a summary of that entire process. So you collect the seeds, they're evaluated and further developed and established in large field grow outs, with the purpose of bulking that seed, in massive production that is then purchased back from farmers who are participants in this program and stored at large on-site facilities that are then used for large-scale ecosystem restoration after fires or other habitat disturbances. And then the cycle continues. But I wanna make the distinction because this program only exclusively targets common native plant species in quantities that can be upwards of 80,000 plus pure live seeds collected in any given season and at a single site, which equates to multi-pound collections with population sizes that are hundreds to thousands of individuals and with all of the maternal lines or individuals sampled being bulked. So none of the seeds sampled from unique individuals is kept separate. It's all placed into one large collection. And you keep track of how many individuals, but not necessarily the genetics contributed by each of those unique individuals. And that's kind of where our work here at the Botanical Research Institute of Texas differs. We focus exclusively on small scale conservation seed bank collections that target rare, threatened, or endangered native plant species in quantities that are as much as we responsibly can. And this is, dependent on a wide variety of factors, the size of the population, how recently it has been collected from in the past, if ever. And the population sizes are significantly smaller, with ideally a minimum of 50 individuals sampled at a given site at a given time. And the maternal lines are kept separate. So what you see here on the upper right is actually one of the tracks in the GPS points of a collection of Rhododon ciliatus that I made, pictured here on the lower left. And you can see my sampling pattern kind of serpentining through the site. And each of those points is a maternal line or an individual that I sampled seeds from for this collection event. And all of those seeds are kept in separate maternal line envelopes. So you can see how this can be a lengthy process, but that allows us to actually keep track of those maternal lines through time. And then all of this to then bring it back to the lab, right? We accession them, allow them to equilibrate to laboratory conditions until their moisture content is the same as our ambient relative humidity and temperature of our space. And this often takes a minimum of 2 weeks post-collection. And then we get to do kind of the fun part, which I didn't have a lot of experience doing when I was a technician with the Seeds of Success program. Once we collected the seeds, we shipped them off, and that was the extent of our hands-on experience with them. Here, because we have such small quantities of seed that we collect from these really sensitive populations, we do all of our cleaning by hand. And you can kind of see in this video that's going on to the left, I am cleaning Areocolon cornicianum, which is a federally listed endangered plant species here in the state of Texas, using a dissection scope and forceps and picks. Because these seeds are really, really tiny. And so the way that we clean things differs as much as the methods required to collect them. And sometimes that does require a little bit of creativity as well. I've even heard, so, the cleaning equivalent of wildly flailing a tennis racket, there has been threshers made out of paint buckets and modified weed whackers, for example. So there's a lot of different methods that can be used depending on the species collected, their size, and the quantity, right? After that, we curate them. So once an accession has been cleaned and inventoried, we split the collection into duplicate base and backup accessions. And so 40% of all of the seeds that we collected will go into our primary base collection, which remains here on site and goes into long-term storage, versus the other 40%, which will be placed into backup base, which goes into long-term storage at another institution. And that is for safety reasons. If for any reason something catastrophic were to occur here, our freezer were to fail, a large, natural disaster were to happen here at our location, we are assured that half of that collection is preserved in some other geographically isolated place where they will be safe and cared for. And then 10% of those seeds will be placed into primary curation, which is used here for research and viability testing. And then the remaining last 10% will go accompany that backup-based accession to wherever it is sent. So after all of that, right, it is time for seeds to be dried down to the final moisture content that they can be stored under orthodox conditions. And this is roughly 25 to 35% equilibrium relative humidity. And this is important because when you place seeds into sub-freezing temperatures, if the moisture content is not low enough, ice crystals will form due to the water content in the cells and damage the cellular structure and cause those seeds to die or become inviable. It'll damage them, right? So we have to dry them down to just enough that when they're placed in freezing sub-zero conditions, they undergo what's called vitrification. And this is a formation of a glass that protects the embryo instead of creating ice crystals that will actually further damage the tissues. So that temperature range was determined to be roughly 25 to 35% for our orthodox seeds. And that's also important because you can over dry something as well. And actually, when you place seeds into sub-zero long-term storage conditions, further drying will actually occur. And so that 25 to 35% not only ensures that ice crystallization will not form inside the seeds, but it'll also ensure that that extra little bit of drying will not be enough to harm the seeds either. And that all brings us to research and development. And you can see here on the upper left-hand corner, a germination growth chamber, as well as seeds on agar that are being scored for their germination on the bottom left. And that's because we use these seeds to study seed dormancy and their germination requirements, because unlike agricultural crops, very little is actually known about the dormancy and germination requirements of native seeds. So that's where we come in. And we use part of these seeds that we have in curation, in those curation packets, to do some of this work. And as a process, or as a product of which, we're able to then develop protocols to share with other institutions and people and folks doing this work to be able to reliably germinate seeds and overcome their dormancy. We're also then able to study seed longevity, because once we place our seeds in long-term storage, we need to be able to check at regular intervals to see how long-term viability of those seeds may potentially be affected by their long-term storage, and just ensure that the seeds that we have and are maintaining here in our long-term storage are still actually viable. So in the event that they're requested back, we know that the receiving institution is getting live seed. And then we're able to also do statistical modeling. And the reason I bring this up is we're actually currently working on a project to develop habitat threat models for sensitive Texas native species to better streamline the actual listing process and identify the threats, habitat level threats that are impacting sensitive native plant populations. And these collections and the data generated from each of these collections really helps us to develop those tools that further the conservation of these species in situ. And so with that, I just want to say thank you to everybody for attending, and I would be delighted to answer any questions or receive any comments. Thank you.
Thanks, Kay, for this presentation. I feel like I thought I knew what went into the process, and it is so much more than I realized.
Yeah.
We do have some questions in the chat, and I also have questions as well.
Oh, fantastic.
Mariah did ask if it was possible to get a copy of the presentation. I let her know that we're recording this, but maybe there's some information in here that she wanted to look over further. But
Oh, sure. Yeah.
I can put your email in the chat.
Yes. Sounds great.
And then Beth has an interesting question. I don't know if you're also looking at the chat too, but do you know if there are studies designed around how long seeds survive in the field? For example, let's say a structure is placed on remnant land and removed later, what viable species might still be in the soil? Or like encroachment from brushy species that provide dense shade for decades before being cleared, which I mean, I know there are examples where this can happen where maybe nothing was growing, some earth was kind of dug up, and now like native plants are starting to grow there. But you know, as far as studies go, and if you're familiar with something like this.
I'm not familiar with something like broadly like comprehensive about like an area and a diversity of different. Most of the studies that I know of are targeted towards like a specific species. For example, the tiny species Eriocolon cornicianum that I was cleaning in that video, um, it it is it needs a certain level of disturbance, but is also a super poor competitor, and also not it's like a Goldilocks plant. It like needs a little disturbance, but not too much. And so like a lot of work has been done understanding how much of that seed um is present in the seed bank, because it's also a very bang and bust type of species. So some years it goes bananas, there's thousands of individuals, and then there's long periods where you don't see one. And so work has been done with that species specifically to look into, okay, how much of the seed is present in the seed bank? Is it viable? And uh, basically as a means of getting at like what's the deal with this bang or bust cycle with this species. Is it that there's not a lot of seed in the seed bank, and then it's like no, there definitely is. So there's some other factor that's impacting the life cycle of the species. And so there's a lot of kind of targeted literature like that, but I would be curious for sure to look into um, what work maybe has been done on a kind of a broader, in a broader kind of lens. But I sadly don't know of anything specific.
I'm wondering if maybe she was thinking of this. And Beth, you're welcome to unmute yourself. Beth helped georeference our specimens from Collin County and found an interesting prairie that is now like built over, or a big part of it. So I wonder if maybe that kind of part piqued her interest on that. But one of, oh, and she asked if there's a link to your video on seed cleaning.
Oh yeah, that will be on Instagram.
Okay.
So I can put my my like little whatever
Handle
it's
in there.
if you use it. This is my Instagram handle.
Perfect. Thank you for sharing that.
And I will
Uh,
say, Beth, really quickly, sorry, that, for prairie specifically, um, prairie the prairie soil has been dug up as a means to more or less inoculate restoration sites, because of the fact that many of the seeds in the seed bank do persist, um, and can be used to help hopefully restore other sites that are being restored in areas where you are seeing that development that is um, impacting, remnant prairie sites elsewhere.
That's interesting. And I have some of the soil from that area that Ashley mentioned was completely forklifted and like tons of it was brought over to the UTD campus in the 70s, but I don't know where
Mm-hmm.
it went. I think it's probably been developed over.
Mm-hmm.
So I know that's been something people have done for apparently decades, which is interesting. But I have anecdotally heard from people and kind of seen like brush clearing after sometimes decades, and the things that are coming up are pretty interesting. But then I don't know, maybe they were there and they were just really small and stunted from lack of light. So I don't know how someone could actually do that kind of a study without intentionally disturbing remnant land, which doesn't seem ethical either to me.
Yeah, I know, I totally know what you're talking about, and I think there has been a number of instances. I mean, seeds are very, very resilient, in many cases. They're built for this purpose of staying in the seed bank, a lot of them, and obviously this is I'm saying I feel like very blanket statements. So also take that with, through view that through the lens of this differs significantly depending on a taxa by taxa basis, right? But, broadly speaking, seeds are very resilient and can survive in the seed bank for sometimes very, very long periods of time, um, waiting for that moment where environmental conditions are such that um, they are conducive to their growth. So things where there's brush cleared or fire has cleared the understory away and exposed the soil surface to light for maybe the first time in many years, right? That has resulted in observations of native species that maybe had not been seen at formerly degraded sites for quite some time, just because of the elimination of that competition, that you're kind of mentioning. So that I don't have any specific um, studies, but certainly, again, like anecdotal knowledge of these things happening for sure.
Hey, I have a question about the connection between herbaria and digitized data with the work that you do. Yeah, how are those connected and how do you use that data?
Yeah, well, certainly, so many ways. So for sure with scouting, scout planning, field planning, knowing where populations have been historically, where they've been recently seen. A lot of digitized herbarium data helps with creating lists, scouting lists. For example, this we're going on a foray with LSU next week, and herbarium data that is on Torch is instrumental to making plans of what has been vouchered, um, where has it been seen, what has been observed but hasn't been vouchered. Like all of these things help with planning of obviously voucher collection, but seed collection as well, and knowing where potential candidate sites we could visit might be, but also for seed collections, collecting vouchers whenever possible is part of our protocol, and that's in part due to increasing knowledge and documenting the occurrences that we sample, but also because we can't know or be sure that something is what we say it is without having that reference voucher. So especially if we're accepting deposits, outside deposits, we want to be able to have some sort of photo voucher or herbarium voucher to refer to in case there's any discrepancy or any kind of contest, basically, of what the identification of that species is, which I've encountered with Seeds of Success, coming upon populations that were collected as something that they weren't. And then being able to pull up that voucher and confirm, yeah, no, it wasn't that thing. That's really important, especially for conservation and things that have maybe very morphologically similar, closely related species that could be easily misidentified. Voucher specimens are really important.
Another question I have, thank you for that. I love that
Oh, yeah.
answer. Of course I do. You may have told me this, or maybe Brooke has told me this before, but what is the max number of seeds we think we can store at this moment? Or how many more seeds? Or maybe what's the
Uh,
total? Do we know?
the total, I know the total. Our current total is roughly 75,000. Probably that's an estimation. We have a little bit that is in processing. So that's just based off of what we previously collected, what I feel comfortable saying. We probably have a 75,000 seeds, and our freezer is only a fraction of the way full. So we have a ton of space still in the equipment that we currently have, and so we definitely have room to expand. I could not begin to estimate how many more we could reasonably accept, but we have 54 accessions right now, and we could easily accept um, 4 times plus this. It's like we've
Yeah.
our capacity has not been met,
Yeah.
basically.
Well, y'all are still in like the baby stages. I mean, do you think that compared to other seed banks in the US, I mean, are we relatively like the newly opened?
Yeah, we're definitely one of the newer kids on the block for
Yeah.
sure. I don't know if we're the newest kid on the block, but we are definitely newer.
Mhm.
And that in a lot of ways is um is nice because well, we're building this from the ground up, and so like our data collection program, our outreach, all of these, our partnerships with municipalities and all of these things, um, we're not having to go backwards and kind of like backwards address huge legacy collections like other organizations are having to now retroactively do. We're kind of able to build for our future from the start, having learned from everyone else. So that's always like really handy, standing on the shoulders of giants, basically.
And
That puts
having
us in a
so many
really,
like protocols that exist out there that
yeah,
you can like use and tweak and all the
yeah,
things.
it
Yeah.
puts us in a really strong position to do this work really effectively from like out of the gate. And so the 54-ish accessions that we have right now, that will grow pretty much exponentially as we're going forward. The next field season specifically is my personal first field season in earnest, and so I will be spending a lot of time this fall putting together a field plan for that, and really collecting in earnest. And so that will only get better with time and with relationships with NIPSOT and NPAT folks and other organizations. So yeah, it's really exciting sometimes to be the new kid on the block.
Definitely. We have a few more minutes, but we have at least two more questions.
Okay.
Someone asked if there's a website for endangered Texas natives for the citizen scientist. And I think TPWD has a website where you can look up by county. Am I right?
Yes, so there's a couple of things. So TPWD has a list that's just published to their website, and then there's our test, which is the website that you're thinking of, Ashley. And I cannot recall off the top of my head exactly what our test stands for.
Rare,
But
threatened, and endangered.
yes, species of Texas probably. I should know this, but anyway, that you can look up by county or by specific species. If you're looking up, the taxa that you're trying to find, it'll tell you what counties it is known to occur in. But definitely that resource I use all the time.
And I finally found the link, so I will add that in here. And then in the PPS, I don't know, Tiana, if you want to unmute.
I've just unmuted.
Awesome.
All right, let's hope I'm paraphrasing this correctly, but we have a question from here. First off, thank you, Kay. That was a wonderful presentation. The question is about, you mentioned the next generation and training and all awareness. And can you give us any advice or steps that, somebody of this next generation, the youth, what can they do to help prevent biodiversity loss? What can you provide any advice or steps for that, that end?
Sure. Mostly, it may sound, I may just be preaching to the choir, but education. Always thirsting for that knowledge to find out what's going on in your community, what organizations are recruiting volunteers, our organization working with us, NIPSOT, MPAT, Native Plant Society of Texas, and the Native Prairies Association of Texas. Both of those organizations are super, super active in the community and education and outreach and growing plants and rescuing plants and all of the things. So I would certainly encourage you to participate if you're able in any one of those organizations. There's lots of great mentorship opportunities, field trips that really kind of get your feet wet, learning the flora and kind of what are the biggest threats to the flora in or the ecosystem in your particular area, and just advocating for the preservation and the conservation of native biodiversity. That's huge, and a lot of that momentum is just from folks who are passionate and interested in educating themselves and realizing the importance of these ecosystems and the organisms that are found within them. So none of that momentum would even be possible without that. So that would be my first recommendation for sure.
Well, I think step one, you've planted the seed of
Yes, I
conservation today.
I hope so. And if you are in the DFW area or Fort Worth and interested in working with us or maybe volunteering with the Seed Lab and learning more about what we do, please feel free to reach out to me directly, and we'll see what we might be able to drum up.
Yay. Thank you so much again. And I think this was a wonderful note to end on. You're getting a lot of praise in the chat. This
Thank
is
you.
so appreciated, Kay.
Thank you for having me. I really appreciate it.
Of course. All right, y'all. Well, we're at 1:01, so I'm going to end this call, but y'all have a good rest of your Thursday. And Armchair Botany will be back in November. All right. Have a good one. Thanks.
Bye, everyone.